Hi, this is Science Hangout from Science Mission. We have today Dr. Matt Bogyo from Stanford University. He is the professor of microbiology, immunology, and pathology at the Stanford University. Dr. Bogyo has won several awards. For example, he is a Searles Scholars Program 2004. He was also a Terman Fellow, Stanford University in 2003. Professor Bogyo is an investigator in pathogenesis of infectious diseases. Burroughs Wellcome. In 2008, Strategic Program for Asthma Research, Dr. Bogyo won Early Excellence Award. He is invited to attend Cavalli Frontier Science Symposium. In addition, Dr. Bogyo has presented or published several articles, more than 150 articles in very well-known publications. So welcome, Dr. Matthew Bogyo.
Thank you. Thanks for having me.
So it's nice to have you on Science Hangout program. Can you please tell us about your background, your education, and some of the projects in brief, what you are working on?
Yeah, sure. I got interested in science. I guess I've always been interested in sort of math and science ever since I was a kid. I have some relatives in my family who are scientists, including my own dad, who's a biologist, and one of my relatives who's a chemist. And then my grandfather was a neuroanatomy professor at MIT. So I was kind of exposed to a lot of science early on, but I always just really liked it. I originally thought I was going to be a physics major. I really like the sort of math aspects of physics, being able to solve problems and get to, number solutions. But then as an undergrad at Bates College up in Lewiston, Maine, which is a really sort of small liberal arts school that I attended, I took a chemistry class and had a really amazing organic chemistry professor, and then just sort of fell in love with chemistry and the idea of being able to sort of make molecules from scratch. And so I switched majors as an undergrad to chemistry, and then finished up at Bates as one of only 5 chem majors they have there each year. And then I went on to do my PhD at MIT in chemistry. So yeah, I've always been interested in synthetic organic chemistry. Originally thought I was going to just make molecules as, become a real synthetic person. And instead I realized that I was more interested in how to apply those molecules than just making them. So I ended up switching over to being more biochem, and actually as a PhD student at MIT, I worked for Professor Hidde Ploeg, who's an immunologist. And so I ended up working in an environment where I knew virtually nothing when I first stepped foot in the door. Like I basically knew no immunology and knew very little biology at the time, but basically learned it trial by fire, and just realized that I could apply what I knew in chemistry to study biological processes. And that's what I've been doing ever since. It's been really a great opportunity for me to think about biological problems using a chemical mindset. And so that's really the direction I've taken ever since. When I left Hidde's lab, I started my own group at the University of California, San Francisco, as part of the program called the Faculty Fellows Program, which basically gives you a small lab and some funds to basically start your own group right out of grad school. So I never did a postdoc. And that was really a great opportunity as well. And basically I started working on studying proteases, in particular proteolytic enzymes. I got interested in proteases because in Hidde's group we studied antigen presentation pathways. So we studied MHC class 1 and class 2 pathways. Both those pathways involve proteolytic processing of proteins to produce little antigenic peptides. And so I got interested as a grad student in the proteasome, because that's the one that produces MHC class 1 peptides. And then the other half of that is the lysosomal proteases called the cathepsins. And that's still what my lab works on, cathepsins and the proteasome. And so, yeah, we've been working in those fields and applying specifically the broad theme of research in my labs to use chemical tools to study proteolytic pathways and other enzymatic targets, in diverse areas of biology.
And do you have any mentors or role models in your career who helped you throughout your growing up?
Yeah, I'd say, for me, the big I had several really good mentors as an undergrad. My research mentor is a guy named Tom Wenzel,
Uh-huh.
who did a ton of research, for such a small, liberal arts school with very small number of chemistry majors. He always wrote grants and got funding, and so I was able to do research starting after my sophomore year,
Okay.
and actually published a paper with him in JAX as an undergrad, which was a big deal.
Okay.
And he just did a great job of promoting undergraduate research, so that was great. And then I'd have to say my organic chemistry professor, Dave Ledley,
Mm-hmm.
was also a huge push for me. He had done his PhD at MIT. He was
Okay.
really supportive of me, and I got to take classes with him teaching where I was one of two students. So you get a lot of interaction with the professors at a place like Bates. And then
Okay.
obviously my mentor in graduate school, Hida Plu, he was always very inspirational, very, very, very smart guy who just had a great sense for what was interesting and just followed his nose in science. And I learned to sort of follow that model, just go after what you think is interesting.
Okay. And have you thought about industry?
Yeah,
And
actually,
okay, you were in industry, right? For a little
yeah.
bit.
So yeah, when I started my lab at UCSF in academia, I was doing a lot of consulting for a company called Axis Pharmaceuticals, which was developing protease inhibitors, and they eventually got bought by Celera Genomics
Okay.
in 2001. And right when they got bought, they asked me to come over and start a group to use some of the technologies we were developing in my lab for sort of chemical proteomics. So I joined Celera just for a short period, and then from there, excuse me, sorry, I'm in the middle of an interview. Can't go. Sorry. Yeah. Okay. Anyway.
Okay.
There's a guy coming in and changing something. Sorry about that. Yeah. Anyway, so what I was saying was that I got a chance to work in industry for a little bit, and at that point, I was able to see what it was like to run a group, and I enjoyed that, but decided that for me, the environment in academia was just I had a lot more freedom, and so that was a lot better for me.
Okay. And can you talk about your lab and your grad students and a little bit about them? How do you interact? What is your modus operandi, so-called?
Yeah, I mean, I guess I learned from my graduate mentor, Hida Plu, is that you have to let people follow their interests, and so I'm pretty open with allowing people to move in directions they find interesting. I have to keep some kind of unified theme in the lab. We can't just work on everything, but generally, get a sense of what people really want to be doing and let them move in those directions. And that's really the only way, people are going to stay motivated for science, because it's such hard work, and a lot of times it doesn't pan out like you expect. And so if you're not really, engaged and interested in your project, you're never going to make any headway. So our group is pretty, broad in terms of the interests, and it's derived a lot by the type of people that come into the lab. I mean, I have a lot of postdocs in my group, so they already have a good sense of research, what they want to do, and so they kind of direct themselves in a way, and that's how it works in a medical school environment. It's less graduate students and more postdocs.
Okay.
That's kind of how it runs.
And what are the challenges do you face every day? Kind of like what do you think makes you think that, oh, okay, I need to go to work? Like how it is challenging to me to get that thing done, something like that. What are the challenges do you have?
I mean, the challenges for all of us now is just keeping the lab running and keeping going. I mean, we're all, as you've heard, I'm sure from everyone, the NIH budget's really very low, have been for a long time, and it's creating a very significant backlog of good proposals into the system. And so, even top work isn't getting funded, and so that's always a worry. The other thing is, because of the way the budgets have been cut, a lot of the 5-year proposals are now becoming 4-year grants, and the budgets are being cut by 25%. And what it means is that you have to write more grants, and you have to write grants more often. So I'd say that's, for me, the biggest challenge. I mean, I like writing grants. It's important to sit down and think about what you want to do. But when you have to write 5 of them a year, it becomes counterproductive. Yeah. So I'd say that's our biggest challenge at the moment. I mean, the science is always good, but trying to keep
Yeah. And what do you do in your spare time? I know you are busy with all the research, writing grants, and all those teaching and all. Like, what are your hobbies? And other things?
Well, I mean, I had. It's funny. I had a lot of hobbies that I had time for when I was younger, and then I have 2 kids now, and so when they were little, you pretty much have no hobbies anymore. You go to work and you take care of your kids. But now my kids are getting older, so I'm getting back into them. I do a lot of skiing and snowboarding, and was really into windsurfing, and haven't been able to do that as much as I'd like because it's very time consuming. But that sort of stuff, yeah.
Okay, and I think let's move on to some of the work you are doing and your presentation. If you could share what you have done, like some of the work you have done, your presentation, sure. I would really appreciate.
Yeah, I can tell you about a story. Yeah. I'll share the screen here, and basically, this is a project that's been going on in the lab for a number of years, and ironically, it goes back to my days as a grad student when I was interested in the proteasome. More for its role in antigen presentation. But this story has to do with the proteasome as a potential drug target for malaria.
Okay.
And so I'm going to tell you some more about that. Let me see if I can get this to move. Okay, so I want to start by thanking the person who did the work, which is really one graduate student named Hao Li, who did virtually all of the malaria proteasome work that I'm going to talk about. We also had a really great collaboration with a group in Cambridge led by Paula de Fonseca. As a structural biologist. Then we've also worked with Anthony O'Donoghue, who was a postdoc in Charlie Craig's lab at UCSF. And then we had a nice collaboration with a group at University of Melbourne, Leanne Tilley. And this work was published this year in February. So basically, we got interested in Plasmodium because it's an interesting organism. It has multiple different life cycle stages. It lives within the gut of the mosquito. You can see there the Anopheles mosquito, and it has a reproductive cycle where it produces new parasites in the gut, and some of those end up in the salivary gland of the mosquito. So when the mosquito takes a bite of the human blood meal, they inject some of these little sporozoite parasites, and those end up in the tissue and move to the bloodstream and end up going to the liver. And once they get to the liver, they penetrate hepatocytes, and inside the hepatocytes they replicate rapidly to produce a blood stage parasite. And then what happens is those blood stage parasites rupture out of the hepatocyte, and they form the blood stage infection part of the life cycle. And this is an asexual stage of the parasite. It basically, all it's doing is finding new red cells and pushing its way inside of a red cell. You can see, I don't know if can you see my pointer at all? Or?
Yeah, I can see that.
Down here, you see one of these little merozoites finding a red cell, pushing itself in, and it forms a little vacuole inside the red cell where it can hang out, and it does that so it can stay away from the immune system. And inside, it starts to undergo these stages where it replicates. It's basically eating a lot of hemoglobin. It gets energy and protein from the red cell hemoglobin. And then eventually it goes through this rapid division process in schizogeny, it's called. And it produces all these little merozoites again, and those rupture out by breaking out of the red cell, and then they reinfect. And so this creates this, every 48 hours, you get this massive sort of replication of the parasite. And then some of the parasites can actually shunt off this way back to the sexual reproductive stage called gametocytes, and those are the ones that get taken up again by the mosquito, and it basically allows transmission from human back to the insect. And as you probably know, malaria is a major world health threat. There's about 200 to 300 million people being infected every year, and now it's about 500,000 people die every year from malaria.
Wow.
So it's a major problem. One of the other big problems with malaria is that it's there's a big issue with resistance, because the parasites undergo these rapid replication stages where they're producing progeny, and they're haploid organisms. They have just one copy of each gene. They can do these kind of rapid shuffling or mutations of their genes to be able to get around drug pressure. And so most of the commonly used anti-malarial drugs that have been around for a long time are now no longer useful because of resistance to those drugs. So what we wanted to get at was, as I said, as a grad student, I was interested in the proteasome. The proteasome is a large protease complex. It's made up of this core 20S particle, which is these rings of 7 subunits stacked on top of each other. So just the 20S has a stack of 7 rings, 7 rings, 7 rings, 7 rings. So it forms a barrel type structure. And then it has these capping groups called the 19S caps, which actually recognize proteins that need to be degraded. And so the thing is it does a ton of different stuff in the cell. It's very abundant. It's found in almost all cells and all organisms. So it's highly prevalent. And it does stuff as important as controlling cell cycle by degrading the cyclins, which are involved in regulating cell cycle. But pretty much all proteins have to be degraded at some point, and they get degraded through the proteasome. So you can imagine it's super important and essential. So what happens in Plasmodium? The idea is if you have something that's really essential for the parasite, it should be a good drug target. And if you take proteasome inhibitors and treat parasites with them, you get this rapid arrest of parasite growth. And what's really interesting is that you can shut down the liver stage, the asexual stage, and the sexual stage inside the insect, all by inhibiting the proteasome, which is really good. Very few drugs out there for malaria can hit all the different life cycle stages. The problem is that if you take currently existing proteasome inhibitors, like for example, there are two anti-cancer drugs for multiple myeloma called Velcade and carfilzomib. These are two proteasome inhibitors that have been FDA approved. The problem is that they hit the human proteasome. So if you try and treat parasites with them, you'll kill parasites, but you'll also cause toxicity to the host. And in fact, if you try and put these into mice, you can't clear infection in a mouse model because the mice get really sick from the drug before they clear the parasite. So we started to ask the question, well, maybe can we get at this selective, can we go after selectivity? Could we find a way to specifically hit the malaria enzyme and not the human? So that's what we set out to do. Again, here's just to remind you the 20S core of the proteasome. It has these different subunits, these alpha subunits 1 through 7, and then these beta subunits 1 through 7, and they're repeated twice in this complex. And of the beta subunits, these are the ones that have the catalytic protease activity. And the red ones here, beta 1, beta 2, and beta 5, all have catalytic activity. They're the ones that cleave proteins that get fed into the cavity. So basically things get inserted into the top of the cavity. Inside they get chewed up into little fragments and then spit out the other end. So basically we want to understand which subunits were important and what happens and which ones do we have to inhibit. So the way to look at specificity is to understand what binds in the protease. How does the protease like to clip things, and how does it recognize certain sequences in a peptide scaffold? So here, this just shows the nomenclature. If you have a peptide that's being cleaved right along this dotted line here, this amide bond, you have certain residues on this side, which are called the prime side, that go into pockets in the protease active site. And on the other side, you have these non-prime sides that have amino acids that stick into these pockets. And so the idea would be that the human pockets here on both sides may be a little different than the parasite ones. We have to understand which side chains fit and which ones do not, and then we can design inhibitors that are selective that way. So what we did was we used a technique that was developed in Charlie Craig's lab by Anthony O'Donoghue. The idea was to take a very diverse set of peptides, and he designed these pools of peptides. He made 124 14 amino acid long peptides. And he designed the sequences such that they had a huge amount of diversity for all possible amino acids next to each other and at position +1 and +2 apart. And basically, because there's a lot of different sites on a single peptide to be cleaved by the protease, in fact, there are 14 on this 14-mer, each peptide has multiple possible sequence opportunities for the protease. And so by using a small number of peptides, you can access basically the entire possible space of natural amino acids that could be cleaved by the protease. So it's a little bit complicated, but basically you're just using the fact that these peptides are very long and have multiple cleavage sites in them to basically create all possible 20 amino acids in each of those positions and allow the protease to tell you what it likes to cleave. And the way you determine what it likes to cleave is you take that whole mess of diverse peptides, you either add no protease or you add protease, and then you measure at various times by mass spec the fragments that form. And those fragments tell you by sequencing what it cleaved. And you can then determine which cleavage sites happen faster than others. And eventually what you get is a sort of a map of what it liked to cleave and what it didn't like to cleave. And we can represent it using this kind of web logo approach. You can see anything above this line here at the top is very good. The protease likes it. Anything below the line are things that the protease didn't like, and the red line represents the cleavage site. You can see in this particular example, which is not the proteasome, but you can see this particular protease liked phenylalanine and methionine in this one position, and sort of liked isoleucine and phenylalanine. It really didn't like small residues like glycine. And so it gives you this sort of zip code of specificity. And so essentially we can do that now with the human and the malaria enzyme. We took the human 20S and the Plasmodium falciparum 20S, this is the malaria one, and we looked for what it liked to cleave, and not surprisingly, we found a large number of sequences that both enzymes cleave very well. But what we were really interested in was this 113 sequences that were more optimally cleaved by the parasite. Okay. So if we just profiled this 113 cleavage sites, we got a specificity map that looked like this, where now we see residues that stick out as being unique. In particular, our attention got caught by this P3 residue, which was extremely large for tryptophan. And you can see this was something that just never gets cleaved very well by the human enzyme, but for whatever reason, the parasite at P3 really likes this. It also has more aromatics, phenylalanine and tryptophan in the P1, which typically are not very well liked by the human. So that gave us the ability to now try and design inhibitors that were selective. And the way we did that was we took an inhibitor that I actually made back when I was a graduate student, which is the leucine, and then it has this what's called an electrophilic trap or warhead group. This is the inhibitor part of the molecule that binds to the active site residue inside the proteasome. And we just basically took a sequence like this, the LU, which was very well accepted by the human enzyme, and we just made those modifications based on our specificity. So we put tryptophan in the P1, tryptophan in the P3, and then a tryptophan P1, P3 combination. And we asked, okay, how does this work? The way we can assess this is to use what are called active site probes. So my group has spent a lot of time developing these things called activity-based probes. They're basically peptides that have an electrophilic warhead on one end and a fluorophore, some kind of a reporter that we can see on the other end. And these are two examples of proteasome probes that were developed by Martijn Verdus, who was a postdoc in my lab when he was working in Herman Overkleef's lab in Leiden. And what you see here is these have the electrophilic trap, they have a peptide sequence, and they carry a fluorophore tag. So when they bind to the subunit that they're inhibiting, you can actually see it because it becomes fluorescent due to the tag. So what that allows us to do now are do experiments like this one, where you have the probe, you add the protease, and it binds. In this case, it will bind to the 3 active subunits, beta 1, beta 2, and beta 5. So if you feed it to, in this case, either the lysate, so this is just total lysate from parasite, or the purified 20S proteasome, you see these fluorescent bands start to light up. And these are the active subunits, beta 1, beta 2, and beta 5. You can see they're marked here. So we can now visualize binding and activity of specific subunits in this complex simply by separating it on a gel. So now what we can do to look at inhibitor binding is to take a small molecule inhibitor, treat the proteasome with it. If it binds in that active site, it'll block the probe from labeling, and you see loss of labeling. So here you see an example where we added an inhibitor to this and this proteasome prep, and you can see it mainly bound this one subunit down here, beta 5. And you can see it completely wiped out the labeling. So we now understand where our molecules bind simply by this competition type of assay. So we do this now over a dose response range where we take, here was the original compound, which is not very selective. It hits both the human and the parasite. And you can see when you titrate it in at about 5 micromolar concentration, it starts to compete away these 2 subunits quite effectively. And you can see it takes quite a high concentration to inhibit all of it in the falciparum enzyme. If we now put the tryptophan into the P1 position, you see we completely eliminate the binding to the beta 5 subunit. You can see we see no effect on beta 5, but we still see the retained beta 2 activity. Tells us that beta 2 accepts fairly large residues, whereas beta 5 does not. If we now put tryptophan into P3, we saw something very interesting. We saw an increase in potency. You can see here, if you compare this lane to this lane, you can see there's less labeling here, meaning that the compound was even more potent against the beta 2 and beta 5, which is good. It means that we're hitting 2 subunits very potently, and we've increased the potency. If we now put the tryptophan in position 1 and position 3, you see we increase the potency. We keep that high potency of beta 2 with activity below 1 micromolar, but now we destroy the beta 5 activity. So we get to a highly selective and quite potent beta 2 specific inhibitor. So we wanted to understand the best way to really design inhibitors and to really understand how this works is to actually see it bound in there. And you can do that by X-ray crystallography. The problem is that it's very hard to purify or express the proteasome because it's made up of all these subunits. So normally in crystallography, you're going to recombinantly express your protein on milligram scale. You'll set up crystals and you get a structure, right? But in the case of the proteasome, you can't do that. You'd have to express all the subunits, assemble the complex, and so you have to basically work from purified material. And it's very difficult to purify milligram quantities of the proteasome out of preps from infected red blood cells. So what we did was we turned to a technique called electron microscopy, which is becoming very, very popular and has really gotten huge improvements in the last few years. In particular, there are much better detectors now available, and there's better mathematical refinement models. And as a result, people are starting to solve structures at high resolution by electron microscopy for big complexes. So this is where Paula came in. I met her at a meeting, and she had just solved the structure of the human 26S proteasome by EM. And I said, hey, you know, this would work for us because we can get away with small amounts of material. So what we did was we took the purified proteasome that we got from the malaria parasite, and it's we only need microgram amounts because it's for EM, not for crystallography. We incubated it with our inhibitor to let it bind, and then you can immediately put that onto a grid and do electron microscopy with it. You can see this is what the grid looks like. Here are our little low resolution structure where you can see the little donuts are looking into the barrel of that complex. And then you can see on the side, like over here, this is one of the proteasomes lying on its side. And you can see now these rings kind of stacked up. But what's really cool is that when you refine the structure down and use some of the algorithms and the new sensitive detector techniques, you can see now actually you can start to pick up alpha helices, and this is actually a beta sheet that you're seeing here by electron microscopy. So we're actually visualizing folds of proteins by electron microscopy, which is really exciting. And you can see down here, we actually got high enough resolution, in fact 3.6 angstrom resolution, and we could get the density of the actual inhibitor bound in the active site. Which was really quite exciting. This is a movie which I don't think it'll play here, so maybe it will. I'll try and play it. Here we go. I don't know if it'll be too choppy, but basically this is the two beta subunit rings of the proteasome. And what you can see here is our inhibitor, our very beta 2 selective inhibitor bound. And in fact, what you see is that it only bound to the beta 2 subunit. It really was selective, as our biochemistry had told us.
Wow, that's impressive.
Pretty neat. And we can now see it bound in the active site. And if I go to the next slide, what you'll see is this is the actual EM structure now, and we can start to understand why this thing was so selective. And you can see here that this is in this S3 pocket where we got so much selectivity. It really likes these aromatic stacked interactions, and it's a very big pocket compared to the and so we now can use that as a way to sort of further optimize our inhibitor. So we're currently working on that. Then the question was, what happens? Can we use this as a therapeutic agent? That's the big question. What's the specificity window here? So what we did was we took, in this case, we used the WLL sequence because it was the most potent, and it had beta 2 and beta 5, which was important for getting good killing of the parasite. And you can see that it was active down below 1 micromolar. We get pretty good inhibition by half a micromolar, so 500 nanomolar. If we put it on human fibroblasts, what we find is that it really has no effect on the beta 2 up until 50 micromolar concentrations, almost no effect on beta 1, and it hits beta 5 only when it gets up into the tens of micromolar. Yeah, so this was good. It means that, and we also knew that beta 5 alone is not toxic to the human host. So if you just hit beta 5, it's generally okay. So we then tested it in cells growing in culture, and you can see this is the kill curve in black filled circle. This is kill curve for parasites in red blood cell. You can see it has about 199 or 200 nanomolar IC50. So at nanomolar concentrations, we can totally wipe out parasites. And this is a short dose. We only dosed this for an hour and then washed it away to sort of mimic a short half-life in vivo. But you can see if you do the same experiment on human fibroblasts, it has some toxicity, but not until about 1,000 times the concentration. So we got about 1,000-fold therapeutic window, which makes us able to now be able to inhibit at a concentration, get the drug into the mouse at a concentration where it's not going to really inhibit the mouse proteasome. So that's exactly what we did. We did a mouse model. You can't use falciparum because it doesn't infect rodents. It only infects humans and primates. And so basically we have to go to a Shibote, which is it's a Plasmodium Shibotii, which is an infection of the mouse. And basically we can infect those, mice. After 3 days, they get pretty high parasitemia in their blood. Then we come in with our drug. In this case, we used the most difficult sort of, one of the big parameters for making a malaria drug is you want something that works effectively with very few doses. You don't want to have someone walk for 3 days to get to a clinic and then be sent off with 24 days worth of pills, which they probably will not take, and that will lead to resistance. You want to be able to give someone a drug one time, ideally, but up to maybe 3 days at most. So we did a scenario where we treated, in this case, IV injected our compound one time at day 3, and then looked at what happened to parasitemia levels in those infected mice. And the way we look at those is we take blood from the mice, we smear it out, and we look by microscopy. And what you see, here's the parasitemia in the mouse, and you can see the green is the untreated mouse. You can see over time, it started off with a few percent parasitemia in its blood, and by day 6, it was up to over 25% parasitemia, and then it sort of stabilizes. But you can see in the treated one, where we only gave it 1 dose of 35 mg per kg of our WLL, vinyl sulfone compound, we knocked down parasitemia to 0, where we couldn't measure it, and then only after 6 days did we see a little recrudescence of the parasite. So this was really good, extremely strong effect, which we were very excited about. And more importantly, when we looked at the mouse body weight as a measure of toxicity, normally with proteasome inhibitors, if you treat them with even low dose of the ones that hit the mouse enzyme, you start to see them lose weight very quickly and they become very sick. Here we saw the mice looked normal. They kept their weight, which suggested we were getting around that toxicity issue. And then one last thing I wanted to mention is that this is another exciting finding that just came out from our collaborator, Leanne Tilley, who published. She showed that what happens is there's a drug called artemisinin. It's considered the frontline drug for treatment of malaria. And what happens, the way this drug works is it's actually a natural product that's found in plants that was originally used as herbal medications in China. And basically what happens is when you take this drug, it's totally stable molecule, but when it gets in the presence of iron, it actually breaks open this oxygen-oxygen peroxide bond, and you form a very reactive carbon-centered radical here. And that molecule is reactive, and inside the food vacuole, it just starts to do damage to all these proteins, and it causes the parasite to die, basically. Okay. It's a nice mechanism because it's fairly nondescript. It's hard for the parasite to become resistant to it because it has a lot of iron around from the hemoglobin that it's eating. Very hard for it to get rid of iron, and it's very hard for it to do anything other than to try and pump this drug out of the food vacuole. So there's been virtually no reports of resistance up until now, and because it's getting so much use, some of the first cases of resistance have just shot up in Southeast Asia. And those are not even truly resistant parasites. They're parasites that have a delayed response, meaning they don't die as quickly when treated with artemisinin. But what Leanne's group showed is that those resistant parasites that have the ability to survive at higher doses of this drug, the reason why they're resistant is because they upregulate what's called the unfolded protein response. The stress pathway that the cell uses to deal with damaged proteins. And so as artemisinin starts to damage proteins, the UPR starts to deal with it, and it needs the proteasome to do that. So what she showed was that, in fact, when you add proteasome inhibitors to artemisinin, you get a synergistic effect, right? You basically shut down the proteasome, you prevent this UPR from coming on, and it prevents parasites from being able to get around artemisinin. And this is exactly what we show here. This is what's called an isobologram, where we're treating both with our proteasome inhibitor and dihydroartemisinin, which is the clinically used drug. And what you see is strong synergism. These dots below the line mean that we're getting very strong synergism, meaning that you can enhance the effects of artemisinin simply by using low dose of the proteasome inhibitor. So that's really exciting because it means we might, first of all, be able to suppress resistance by simply layering a proteasome inhibitor at low dose. And it also suggests that even without killing parasites via the proteasome mechanism, you can enhance their ability to be killed by artemisinin. And nowadays, all drugs are used in combination. So we think that this has real potential for extending the life of artemisinin, which is one of the most clinically relevant and important drugs currently in malaria. And so that's why we're very excited. So basically, to summarize this project, what we really believed a long time ago and now have finally shown is that the proteasome is really sufficiently different from the human enzyme, and we can use that as a way to selectively target. And then we can actually use the sensitivity that Plasmodium has to the proteasome inhibition as a way to really effectively clear parasites. And then we think that this artemisinin component is going to turn out to be really important. It's going to allow us to potentially come up with strategies to prevent resistance, and also hopefully prevent artemisinin from becoming like a lot of the other drugs that's just no longer valuable.
Yeah.
So that's sort of one of the projects related to more pathogenesis. My lab works on Toxoplasma gondii, which is a related parasite pathogen. We do work now with pathogenic bacteria, in particular Clostridium difficile, and we're starting to move into some other bacterial pathogens as well. So definitely part of my lab works on pathogenesis. The other part of my lab, how much do we have enough time? Do you want to hear?
Yeah, we have some more time. Yeah, at least another 15-20 minutes.
Okay, yeah, so I'll quickly, the other thing that we're interested in are these lysosomal proteases called the cathepsins. They hang out predominantly in lysosomes, but they also play some roles in the cytosol and even in the nucleus of cells, and they get secreted and play roles extracellularly. And the reason why I've been interested in them is because if you look at every type of major cancer, solid tumor, breast, melanoma, lung, colorectal, prostate, and glioma, these all have upregulation of cathepsin activity, both at the protein level and at the activity level. And they seem to be associated with immune cells, so predominantly macrophages produce these proteases. And so they become good biomarkers for inflammation, which is an important process for almost every type of major ailment and disease that's out there involves some component of inflammation. And even in cancer has a big component as well. So we started to reason, could we target these things? We use these kind of activity-based probes that I just described. They're covalent labels. Here's the thiol from the cysteine protease. What you do is you design a molecule that looks like a substrate. It gets attacked by the thiol, and you get a permanent bond that forms here, this thioether bond. And so you now have a way of tagging the active enzyme, much like what I showed for the proteasome just a minute ago. In this case, though, we have a leaving group called an O-acyl leaving group, or this is called an acyl oxymethyl ketone, but this O-acyl piece is gone. It gets lost when the bond is formed between the enzyme and the substrate. So what we designed were these kinds of covalent labels that carried a fluorescent reporter and also a quenching group. So the idea is now, here's an example. When it's bound in solution, this is actually an animation, so you can't really see it here, but when it's bound in solution, the molecule is quenched, but when it binds to the active site, the fluorophore turns on because this is the quenching part of the molecule gets lost, it diffuses away. So what it allows us to do is to dump this molecule onto live cells, and wherever there's active protease, it gets bound by our probe, and the probe turns on, and you now see this fluorescent signal. So you can see here, these are macrophages in culture, and these little dots of color are the active proteases inside the lysosome. So we can now actually visualize that. The other thing is you can put it into a whole animal because the molecule can be made fluorescent with a high wavelength dye that's visible through skin and tissues. Here's a molecule here where we have a quencher out here, a fluorophore here. We inject it into a mouse that has 2 small tumors growing in the mammary fat pad. And you can see very quickly within a couple of hours, it accumulates and turns on where there's active protease in the solid tumor. And you can now visualize these tumors. And you can now see that this allows you to quickly detect where tumors are. And this would obviously very much help in deciding how and where to cut out tissues during a surgical procedure. The other thing is because the molecule is permanently bound, we can take the tumors out of the animal and run them out on an SDS page gel, just like I showed for the proteasome. We can see what they bound. In this case, we bound these 4 active lysosomal cathepsins, X, B, S, and L, you can see here. So what we've been doing now is moving from the sort of mouse imaging that I just showed you, where you put a mouse inside a very sensitive system like this that has a camera, it can see the fluorescence on the mouse. That's great if you're a mouse, but if you're a human, you need something else. So what you can use, we've been working with Intuitive Surgical, which is based out of Sunnyvale, kind of close to us here at Stanford. They have a system called the Da Vinci surgery system. It has a camera, and it's basically a robotic system where the surgeon sits across the room and looks into a 3D image and performs laparoscopic procedures inside the patient using a robot. And it's pretty neat technique. It was developed originally by the military by DARPA actually, to try and develop a robot that could go out into the battlefield and do surgery without having to endanger the surgeon. It never quite got that far, but it's being used now. There's about 3,000 of these robots worldwide, so most hospitals have them. And it's allowed new applications in laparoscopic surgery that weren't previously possible, and that's done a lot for helping with patient outcomes and amount of time that people spend in the hospital, because procedures are now done laparoscopically that weren't previously not done that way. So it's really exciting. They also have an attachment here for this camera called the Firefly detection, and it can see fluorescence. So for us, that's great. It means here's a clinical robot that's already being used in the operating room, and it can see the fluorescence that our molecules have.
Yeah.
So what that allows us to do now is it allows us to take one of our mice. You can see here this poor little mouse being operated on by this giant robot that's meant for a human. But basically, we can bring it down to Sunnyvale, attach it to the robot. We can do surgeries and use our probes that detect the tumors as a way to highlight where to cut.
Yeah.
And so what we did is we took one of these mice at about 4 hours that has really bright signal in these tumors. You can see right here. And we just do surgery using the robot. And hopefully this will work. You'll see here's one of our surgeries, and you're going to see this is the image looking down through the camera system. You can see here very clearly the green is the fluorescence. We can adjust the intensity. And it could overlay that over a white light image. And now we can very clearly see during a surgical procedure what's fluorescent. These are the two little devices that you'd be using during the surgery. This shows a very crude resection of the primary tumor. And what you're going to see is in the tumor bed behind the primary tumor, which was very clearly fluorescent, there's another residual patch of fluorescence you can see right here. Fluorescence is residual tumor, and now we can see that because it's lighting up, and now we can go in and cut that out. And you can see without any fluorescence, you can't see that very well at all. It just looks like tissue. But we clearly have some residual cancer. When we went and cut this piece out and analyzed it, we were able to confirm that, in fact, it was a tumor. And you'll see when we remove it from the mouse and put it over here, this piece is fluorescent right here, very clearly.
Yeah.
We've also been applying this to other types of cancer. The one place where we think this also has real value is in colonoscopy procedures, where currently there's no contrast for detecting early stage polyps, which are very flat lesions and hard to see. So by being able to light those up with some kind of fluorescence, you'd now be able to quickly determine where to take tissue samples or where to remove early stage polyps, and hopefully get a better outcome in terms of survival. And here's some example. This is just a mouse where it's been genetically engineered to have these polyps in its colon. We've resected a piece of its colon, and we're using the Da Vinci system just to image it. Right here, we're looking on the outside of the tissue, so you can see the polyps lighting up as little fluorescent spots. If we now open this tissue up, in a minute we'll splay it open, which would be more like what you'd see if you were doing a colonoscopy procedure. Would be inside. This is now inside the tissue, and you can see very clearly these little lesions that are forming, including very small ones that would be very hard to see. You'll see in a minute we'll move down further, and you'll be able to see some that are very difficult to pick out. Here, this is a very large polyp, obviously very fluorescent. Also, you can see some by white light. Here you see some fluorescent, not some stuff by white light, but over here there's some stuff going on that you can't see right here. This is all early stage polyps that you can see just doesn't show up very well by white light. So it allows you to really see where to look and where to perform these resections. And this last few slides just show, that we can take those tissues out, do histology to confirm they are in fact tumors. Here's some of the polyps that we removed, very cleanly fluorescent, and you can see these are definitely by H&E staining tumors. And then I just want to thank everyone in my group. We've had a really nice mix of postdocs and students and people with different backgrounds. And it's been really a good opportunity to work with people with lots of great experience. And, we've had a bunch of really good collaborations. So, yeah, that's about all I wanted to talk about.
Okay. So it's a great presentation. It has a lot of implications for human treatment for human diseases like malaria, as well as for the whatever you showed at the end was really, it's amazing. If you can do that, there will be less recurrence of the tumors in many of the cancers.
Yeah, I think we're currently at an interesting time for these types of optical contrast agents, because the problem has simply been that we haven't had any approved by the FDA other than simple dyes like indocyanine green. And so that only allows you to look at vasculature and other things. But these targeted dyes are really exciting. And there's a couple companies now that have dyes that are entering into phase 1 trials. And I think it's only going to be a matter of time before a targeted dye gets approved and becomes part of standard practice whenever you go in to have surgery to remove tumors. You put something in the patient to light up where you want to cut.
And how long do you think, like, the whole thing, like, I mean, like, from the approval to the market, how long do you expect it to take? Like, within a few years?
Yeah, I think in a few years, because as I said, there's a couple companies now that are in phase 1, and they've already done studies in patients with tumors.
Okay.
To confirm. The hard part is the expense, right? I mean,
Yeah.
To get it through phase 2 and phase 3 trials that the FDA is going to require, you're talking tens to even hundreds of millions of dollars.
Yeah.
And companies just don't quite know what the market's going to be for these type of agents. So it's sort of hard to invest hundreds of millions of dollars in something that you don't know what it's going to make back. I think companies like Intuitive could lead the way because they generate so much revenue from the robot parts that they could actually invest in it. And for them, if it expands the use of the robot, that's good. You don't actually have to make a ton of money off the contrast agent itself. So I think we're going to look to companies like Intuitive to kind of make those steps, and they're doing it. So they are pushing multiple things into the clinic now. So
okay, that's good. And regarding malaria, like, what stage, like, are you getting into the clinical trials or anything, some of your compounds?
Yeah, so right now we are moving these things. We're optimizing them so that we can essentially improve upon the properties. And I think part of the biggest issue for us is, as I said, for malaria, you want something that works very effectively on a few number of doses. The other thing is it has to be orally available.
Yeah.
Currently, the lead compounds that we tested, we don't think they're particularly orally available. So we're optimizing those. And if we can show oral distribution and we can get the cost of goods down for making the molecules, the idea would be to get some organizations like Medicines for Malaria Venture or MMV to pick this up, and with the help of other companies that want to develop new anti-malarials, and we'll see. That's the hope.
Okay. Yeah, I think it's a really, like, it's a really eye opener for me. Like, I had seen some of your papers, but like to learn more in depth from your presentation is really interesting.
Great.
Thank you, Matt. So I really appreciate you joining Science Hangout and taking time from your busy schedule. We'll really love to have you back whenever you have some more good news from your research work.
Sure. Okay. No problem.
Thank you so much.
Okay. No problem.